Hi everyone. Sorry about my scratchy throat today, but I'll try my best to sound as clear as possible. On behalf of our team, I'm going to talk about the research projects that are currently undergoing in our lab with a vision to advance the therapeutic pipeline that is meaningful for the families and individuals affected with Kifani-associated neurological disorders or CAND. CAND is caused by pathogenic variants in the kinesin family member 1A or KIFAN-A gene, where affected individuals suffer from a combination of various clinical symptoms, including severe intellectual disability, development delay, autistic behavior, and epileptic seizures. Epilepsy is a disorder of brain characterized by recurrent seizures. A seizure is usually defined as a sudden alteration of behavior due to temporary changes in the electrical functioning of the brain. Severe epilepsies that begin soon after birth can affect the normal development of the brain and can result in development delay and severe cognitive impairment. We now know that at least 42% of CAND individuals suffer from uncontrolled treatment-resistant seizures. There are no specific therapies that can either reverse or halt disease progression. Although KIFAN-A is known to be a molecular motor protein transporting synaptic cargoes to the tip of the neurons, little is known about the role of KIFAN-A in maintaining normal neuronal network activity. Even less is understood about how associated pathogenic variants lead to life-threatening seizures in children. To address this gap in knowledge, we worked closely with kifani.org to design a project with an aim to improve our understanding of the functional consequences of pathogenic KIFAN-A variants and the mechanisms behind the severe seizures, to identify targeted therapeutic options for CAND children. We have assembled an expert team of molecular biologists, clinicians, and bioinformaticians to characterize the cellular and epileptic phenotype of KIFAN-A variant patient neurons and brain organoids, with an ultimate aim to identify small molecules that can reverse the seizure phenotype in CAND children. Brain cells communicate via electrical signals, and any disturbance in the normal brain activity causes epileptic seizures, which is measured routinely in a clinic using a method called EEG. Small metal discs or electrodes are directly attached to the scalp, and the brain activity shows up as a wavy line on the EEG recording. Seizure activity in CAND often occurs at night and could go unnoticed during a routine daytime EEGs. So the aim of this study is to develop a lab-based epileptic model that replicates the EEG seizure phenotype of affected individuals. The clinical EEG reports from CAN individuals with 6 different KIFAN-A variants will be collected. Induced pluripotent stem cells or iPSCs can be generated from affected individuals' skin or blood sample and essentially can be reprogrammed to a stage where we can force them to make any cell type of the body, including neurons. 6 different iPSC cell lines harboring specific patient variants along with their isogenic controls will be differentiated into cortical brain neurons to characterize the key differences in phenotype of iPSCs derived neurons between CAD individuals and their isogenic controls. We will also characterize the phenotype of iPSC derived brain organoids that are mini brains in a dish that can be grown in special bioreactors using patient-specific iPSCs, and they resemble the architecture and function of the human brain. The neuronal network activity in affected individual-derived organoids will be measured using multi-electrode arrays or MIAs. That is a special device that contains multiple microelectrodes to record neuronal activity in a dish. Using published bioinformatic algorithms, we will compare patient EEGs with their cognate MIA data. This information will define the electrophysiological features shared between EEGs and their cognate brain organoids and advance our understanding of each patient's functional electrophysiology, potentially allowing us to predict the epileptic course in individuals with specific mutations. Most importantly, this in vitro seizure phenotype will provide a readout for therapeutic screening of candidate compounds. For drug screening, high-throughput technologies will be used that we are currently developing in our lab with the kind support of kifani.org. COR7-based cell model stably expressing truncated KIFAN-A harboring A variant that shows the maximum differences in the MIA epileptiform will be used to screen a library of FDA-approved drugs. The effect of the top 5 hits will be assessed via phenotypic characterization, including rescue of neuronal phenotype and trafficking of fluorescently labeled Kifane cargo such as BDNF. To further characterize the validated small molecules, transcriptomics and proteomic analysis will be performed to identify key cellular pathways altered by these small molecules. Finally, we will treat Kifane variant organoids with the top 3 small molecules, and the epileptiform activity of the organoids will be measured before and after treatment using the MIAs. The outcome of this project is to develop a seizure phenotype prediction model and to create an efficient way of identifying targeted treatments for CAN individuals with refractory seizures. Understanding these seizures will enable a more targeted approach to identify seizure-specific treatment options for individuals with CAN and other epilepsies, and will have a dramatic impact on the quality of life of CAN individuals and their families. We would like to acknowledge our collaborators and partners who helped us all the way to design this important project. Special thanks to all the organizations including NHMRC, Kifane.org, MCRI and the University of Melbourne who are supporting us financially to pursue with this study. Above all, thank you to our relentless CAN families for their continuous support. Thank you so much.